The question is, how did Hershey and Chase differentiate between DNA and protein in their experiment while proving that DNA is the genetic material? Now, to answer this question, we need to first understand what their experiment was. So, Hershey and Chase worked to discover whether it was protein or DNA from the viruses that entered the bacteria in their experiment. And this particular experiment led to the proof that the DNA is the genetic material and it is not protein. So let us understand the experiment now. So Hershey and Chase worked upon bacteriophages and E. coli. And also they used different radioactive isotopes to label DNA and protein code in the virus, that is the bacteriophage. Now if you look at this figure, there are two bacteriophage present here, and the bacteriophage is a virus that attacks the bacteria. And in this experiment, what they did was they radioactively labeled protein capsule in one bacteriophage and they radioactively labeled DNA in the other bacteriophage. And here in this case, the radioactive element was 35 sulfur, whereas in this case, it was 32 phosphorus. Now, once these bacteriophages were radioactively labeled, they were injected into the bacteria. Then the process of blending occurred and then centrifugation took place. Now, once centrifugation took place, the results and the observations that were incurred were that no radioactive, that is S35 radioactive, was detected in the cells, but radioactive, that is of 35S, was detected in the supernatant in the first case, in which the protein capsule was radioactively labeled. So here in the supernatant, the radioactivity was seen. Whereas in the second case, the radioactive phosphorus was detected in the cells, but not in the supernatant. So from these observations, they concluded that since the protein coat was lighter, it became the supernatant, while the injected bacteria got settled at the bottom. So in the first case, then we can say that the radioactivity was seen in case of the protein coat which settled down in the supernatant and not in the cells above. But in the second case, the radioactivity was seen in the cells and not in the supernatant. So hence, it was proved that the DNA is the genetic material, as it was the one that got transferred from the viruses to the bacteria and not the protein, because proteins remained in the supernatant and were not transferred to the cells, because if they would have been transferred, they would have been seen in the cells, which did not happen in the first case. So this is how Hershey and Chase experiment led to the conclusion that DNA is the genetic material.